In this video, I want to cover the second part of the assignment, where we have a look at DNA molecules. I just need to find a pencil. A pen will do. I have 4 different nucleotides that make up DNA: adenine, thymine, guanine, cytosine. Adenine always pairs with thymine. There are 2 hydrogen bonds. Notice this is a nitrogen, electronegative nitrogen bonded to 2 hydrogens. That means those hydrogens have a partly plus charge, whereas the oxygen has a partly minus charge. That's why those 2 stick together, just like a water molecule. Same thing here. I have a nitrogen and I have a hydrogen connected to a nitrogen, which causes another hydrogen bond to form. Adenine and thymine have 2 hydrogen bonds. Guanine and cytosine have 3. Looking at this, I can tell, same thing. This is my cytosine. It has 3 bonds, so it either has to be a guanine or a cytosine. This is my reference. Looking at this, I can tell this matches 3 hydrogen bonds, 2 rings. That's another guanine. This has 1 ring, 3 hydrogen bonds. That's cytosine. Excuse me. Adenine has 2 rings. Here's 2 rings with 2 hydrogen bonds. That's an A. And then thymine has 2 bonds and 1 ring. Those are my letters. These are the 4 letters in DNA. DNA has A, T, C, and G. In RNA, you have something called uracil, which replaces the T. So in RNA, you'll have A, U, C, G. I will talk about that in another video. From there, DNA is a double-stranded molecule, which is why I asked everyone to have 2 strands. And then the task is to change up this code, but anytime you change up the code, A must go with T and C must go with G. So if I quickly cut this up, and I don't need these names anymore. Cut through the middle and separate the nucleotides. This would be a little bit of dehydration synthesis. I can do that with my other molecule. I don't just want 4 things on a side. It'd be nice to get a longer chain. And again, if I'm really careful in how I look at this, even without my names, I should be able to figure out what I have. I want you to get some experience looking at these things and trying to figure out what they are. So my G has 2 rings, 3 hydrogen bonds. That there is a G, which means 3 bonds. This is also a G. My C is 3 bonds with 1 ring, so that's my C. That's my C. My A, 2 rings, 2 bonds. Here is my A, and I have another A here. And then my T is 1 ring, 2 bonds. So with that, based on my knowledge, based on comparing, I start recognizing what these letters are. And these are the letters for the code that makes everything inside of living organisms. This is the code that directs how an organism is made. If you master the DNA code, there's really no stopping what we're able to do in terms of making life do what we want it to. This is the whole premise of Jurassic Park, among many other Gattaca, many other movies that deal with genetic engineering, or the whole controversy, or really needless controversy about genetically modified foods. It's all about manipulating the DNA code. So we have our 2 strands of DNA. If I start with a G on one side, I'm going to need a C on the other side. This might be too bright to be able to see what I'm doing. Turn down the brightness a little bit. It's a little bit clearer. Now, if I put down a T, and it should be on the, this is where the phosphates go. On T on one side, I should have an A on the other side. If I put a G on this side, I should have a C. Which I'm looking through my samples here. So a C should match up. Here's a C. And then let me just randomly pick another A and a T. How would I change the code? Well, I could switch these up. Really, I only wanted you to give me 2 separate codes. Now we see A to T, G to C, C to G. I could also switch this in. I could put a G here and a C on the other side. These are all possible codes. Always the code. On one side, AAGG, the corresponding TTCC would be the reverse strand. This is how the DNA molecule stays together. And this is the code. We'll see. This is the code that drives how an organism does pretty much every process within its body. So really all you need to do is give me 2 different chains with 2 different series of sequences. So AT, GC, GC. I could do AT, GC, GC, A, and then a T. The important thing to remember here is that the A's and the T's always match up. So I can't have an A to an A, for example. It has to be an A to a T. Where's my other T? Well, this A would join to that T. Phosphates are on the outside, that PO4. The nucleotide, the nitrogenous base is on the inside. That's what does the hydrogen bonding inside the molecule. So that's your assignment, and please send me the pictures. Thank you.